So Alex has asked me to give this talk. It'll start to, when I start, it's going to seem like it doesn't relate to vaccine, but at the very end, hopefully we'll relate it to vaccine a little bit. So this was a study we've done several years ago, and to look at the effect of Pap smear collection on infection in a rhesus macaque model. And quite frankly, I thought that this would cause more controversy in the gynecological field, but since the years we published it, it's basically been ignored. I think that maybe clinical gynecologists think it's just some weird animal model. It doesn't have any relevance to the real world. So I'm going to go through a little bit of the data from this study, and you can decide for yourself whether it's relevant or not. So the next slide. So this story starts when we developed a mouse cervical vaginal challenge model that Margaret briefly mentioned. And the finding shown in red here that I want to emphasize is that in order to get infection in this model with our HPV pseudovirus, we needed to have physical or chemical disruption before we added the pseudovirus. And so we looked at why we needed to have disruption. And next slide. What you see here is if you put in particles that are labeled with a green dye but are still fully infectious, that if you have undisrupted epithelia, there's absolutely no binding to either stratified squamous or simple columnar epithelium in the cervical vaginal tract. Next slide. But it was amazing observation when we saw this, that as soon as we disrupt the epithelium, immediately we get very strong and only binding to the basement membrane that separates the dermis from the epidermis. And it's only after about 24 hours that in the middle panel, while it shows up, you can see transfer to the cells. And then at 48 to 72 hours, do we actually see infection? Next slide. And again, Margaret went through this, but just start clicking through it. You can see that what we think is happening is that the virus first binds to specifically modified forms of heparan sulfate proteoglycans, and then this causes a conformational change, which exposes L2 to furin, which in turn exposes a surface on L1 that can now bind an L1 specific receptor on the keratinocyte. And for the next slide. So the implication for this study is that interventions that disrupt or permeabilize the epithelium to the extent that the virus can access the basement membrane are by our model going to potentiate HPV infection. Next slide. So we were struck by the fact that Pap smear collection disrupts cervical epithelium by design. And so we decided to ask the question whether potentiation of HPV infection occurs after collection of a Pap smear. We couldn't do this in women, so we turned to a non-humate primate model, rhesus macaque specifically. Next slide. So this is a study that was done several years ago here. It's listed there by Jeff Roberts. And I would emphasize that he had completed his residency in OBGYN, so could collect a Pap smear in the monkeys just like he did in women. And so this involved a speculum exam, either with or without cytology collection. And we used a standard cytobrush for endocervical collection of cells and a spatula for ectocervix. And then after doing that, we instilled HPV-16 pseudoviruses that express the red fluorescent protein so we could monitor infection events as individual infected cells. And then Jeff did a standard digital exam, and he did it by using one of two lubricants on his glove. One Surgilube, which is the one that's normally used, at least here in the United States, or he used carrageenan, which I think many of you know is an HPV infection inhibitor, which we had shown is a very strong inhibitor of infection both in vitro and also in the mouse model. And then 3 days later, we sacked the animals and excised the reproductive tract and then sectioned it, and then basically on slides counted the number of infected cells by confocal microscopy, looking at over 600 images and two separate counters doing this blindly. And the way this was designed was that there was three groups of 4 monkeys each. In group 1, there was pseudovirus that were distributed achromatically. In other words, there was no pap smear. In group 2, we instilled the pseudovirus after doing a pap smear, and then did the digital exam using Surgilube on the glove. And then as an intervention, we did the same thing, only the only change was that carrageenan was on the glove rather than Surgilube. Next slide. And this is just sort of the actual data that at H&E of the ectocervix transformation zone and endocervix. And I hope you can appreciate in red that there's these infected cells in the presence of challenge after cytology, but not without cytology. Next slide. And these are the numbers, and the results were not subtle. So if you look at putting the virus in without any pap smear, there was basically no infection. We could barely detect any infection events at all. With Surgilube, there was more than 1,000 times more infectious events. So something like 85 infection events. You can see the spread. And almost all of those were interrupted or prevented if we simply made the intervention that on the glove, the digital exam was done with carrageenan rather than Surgilube. So these are really quite dramatic results. And just to note, we did see infection using this of the ectocervix, the transformation zone, and the endocervix. And it didn't look like the transformation zone was exceptionally susceptible to infection as opposed to the other sites. So next slide. So what are the implications of these results? Well, first of all, when we wrote this paper, we wanted to make it really clear that this wasn't a call for changing pap smear recommendations, because it's really clear that the increase in susceptibility, first of all, is going to be transient. The epithelium repair is probably within a few days, and that it's eminently clear that organized screening programs clearly decrease the rates of cervical squamous cancers. But one of the questions that are raised in our mind is, what about the unexplained increase in the rates of adenocervical carcinomas in young women? And just to show you a couple bits of data, next slide. So this is some data from the United States published in 2004, where if you look on the left, you can see the rates in blacks and women for squamous carcinomas are decreasing, but there's this marked increase, particularly among white women, younger white women, in the incidence of adenocarcinoma. And next slide. Just to give you one other example, this is more recent data from the Netherlands, where again, in women under 40, you can see that there's a substantial increase in the rate of adenocarcinoma, which to my knowledge has still remained unexplained. So one of the questions, I think, next slide, I keep advancing it for my thing. So the question then is, does more aggressive collection of adenocarcinoma cells, for instance, with this cytobrush, which had been implemented during this period, does that promote adenocarcinoma at the cervix? In other words, is there an association between the increased frequency and aggressiveness of pap screen collection and the subsequent rate of cervical adenocarcinomas? The idea being that if younger women have ectopy and you may be sampling cells that are younger in areas that now are going to get infected and then recede up the endocervical canal, and then later in life you're no longer going to be sampling those. So we're basically potentiating trauma to the endocervical cells in areas that otherwise wouldn't be traumatized and subject to infection. So next slide. So the question raised, some questions maybe to discuss the implications of this study and what to do next is we've sort of thought about whether we need to conduct a trial to compare the rates of HPV infection with pap smear plus or minus carrageenan. And don't forget, so the carrageenan was just put on the glove. We made no attempt to actually take some carrageenan and place it, for instance, at the cervical os after doing the pap smear to try to inhibit infection. Another thing you could say, well, why don't we just change and use carrageenan gel rather than Surgilube? Because carrageenan gel is marketed as a sexual lubricant, so there wouldn't be any reason why you couldn't just use it essentially off-label rather than Surgilube. Another thing that I think that was raised earlier, I think maybe by Alex, is if Pap smears potentiate infection, what does this say about our natural history studies where we're measuring rates of acquisition of HPV by doing repetitive Pap screening or Pap tests, essentially by doing cervical scrapes? And should we be doing this more by atraumatic types of mechanisms for HPV DNA testing? And in terms of public health, is this another reason why we should be going to DNA testing that, for instance, collected by self-sampling rather than scraping the cervix every time we want to determine if somebody's infected? And finally, the last thing, and this is the thing to do with vaccination, it raises the possibility whether vaccination of mid-adult women could prevent this auto-inoculation of the endocervix after Pap smear, and thereby may reduce the rates of cervical adenocarcinomas. I think this is a big question. I mean, I certainly wouldn't want to push this too hard, but at least it raises that this could be sort of an unthought of or not widely appreciated extra benefit of vaccinating mid-adult women. And so with that last slide, I'd just like to acknowledge my collaborators, especially Jeff Roberts. Some of you may know Jeff. He's now at the FDA, and he's actually quite prominent in reviewing the HPV vaccine trial stuff. But he originally was in our lab, and like I say, he was a trained OB/GYN and well suited to do this particular trial than all the rest of the people in the lab. So that's it. Thanks.